Okay, guys, if you can hear me on the internet, so guys, if you can hear me, please give me a smiley face. Again, if you can hear me, can you give me a smiley face, please? Okay, so apparently I'm going to try this. Can you hear me? Can you all hear me? Okay, I'm not a good typer. Real, right? I'm not a good writer either, so we just kind of go with whatever I have, which is not very good right. So hopefully you can hear me. We'll say yeah, probably good. Let's see what hopefully she said yes. Perfect. Awesome. Okay, so what we're going to do is it's going to be week 10. Biology, what we're going to be doing is I want you guys in the internet to basically, you're going to be turning in the essential question I'm going to give you on the next slide, as well as I'm going to go walk through the tutorial on how to actually get the answer. Because the answer is going to be part of your lab, because the lab you should have technically done if you're not here, I believe yesterday or 2 days ago, I think it was Wednesday or something. So some of the people here, they just didn't do anything that day, or they worked ahead, or they were doing science notebook, or they slept. Right? That's a good answer. And so what we're going to do is we're going to go over that tutorial. After we go over the tutorial, I'm going to go ahead and kick you guys off. We're going to focus on getting done here, and they're going to double dip their points. They're going to get points today for showing up, as well as they're going to get points for their lab. So good for them. All right, so how's it going? By the way, science notebook should be about halfway done. This is week 10. We have approximately 20 or so weeks in the semester. So your science notebook should be going. If you have no idea what I'm talking about and you didn't watch my orientation video, for week 1 or week 2, or look on your computer at all on the left-hand side, the very bottom left, where it says science notebook. If that makes no sense, you need to do that because there's about 100 points. You can even get extra credit by doing that, which is good. So actually, I think I gave up to 115 points to some people because it was just amazing, so I had to give them extra. So make sure you do that. And that's about it. Let's go here. Okay, so the essential question is that what I want to do is how do each of the test tubes, all right? Some of I just typed it right now, so I made just a few grammar errors. I didn't read through it. Make sure you always read through your stuff before you submit it. But how do each of the test tubes react with the egg, and then why do they react in the way that they do? So what we're going to be doing is you're going to be taking 5 different vials. Well, we can do it 2 ways. We can do it virtually, where we can actually use the chemical and see it, or we can use the test tubes with stuff like pepsin and like acids and stuff that if you get it on your face or anywhere near you, burn you. So I figured, instead of you guys burning yourselves today, today. So we'll do it this way. Plus, when we do it this way, we actually get true reactions. We don't have to make sure there's, like maybe there was something in that vial before that wasn't washed out properly. We get rid of all of those anomalies. So the eggs will react, or the protein will react in a certain way. So let's go ahead and look at that. I'm going to share application. No. Virtual lab. Okay, so I'm going to go ahead and restart this lab, and I'm going to go and show you guys, go through the tutorial, and we're going to see how to do this. And then we're going to get rid of the people online so they can go off and do it on their own time. And the people here, we're going to quickly go through it and get all the stuff we need to get done. Deal? Deal. Okay, real quick, if you want to make it larger, right here. If you want to lock it down and not so it doesn't scroll and move around, right here. If you want to adjust the speakers, right there. This is if you want to zoom in or zoom out of the lab. If you need to look at some things, there you go. If you want to leave the lab, right here, the little home is a reset button. Okay, so the first thing we need to do is get the protein that we're going to be analyzing. And what we'd have to do is here, we'd have to have eggs, and we basically boil the egg, and that's basically pure protein if we take the yolk out. So we go over here, go down, we take the egg. Now look how amazing this is. Look at that. I took the yolk out, I cut it up. Look at that. So fast, you can see it. And then we pick up the spoon, and what we're going to do is we're going to put these in the 5 different test tubes. So here we go. 1, 2, 3, it down. Okay. Now we're going to move over to the granulated cylinder. We're going to get 10 milliliters of water from the H2O. So there's the 10. I'm going to put it in the cylinder. Now I need to make sure, this is the part where some people mix up, I need to make sure I put 10. Each time I put it in there, it only goes, I think, it goes to 2.5. So I do it 4 times. Now I have 10. I'm going to go ahead and put this back as soon as it allows me. Okay, I'm going to take this. I'm going to go ahead and pour it into the very first test tube. Now this is the weird, you have to like kind of wait till it gets to the point where it'll turn over. It is done, so I set it down. This is where this got mixed up, because after I stirred it, I thought the computer like broke. So it took me a couple times to go through. That's why I figured I might as well go through this with you. After you're done, you need to go put it up here on the use stir rod. I didn't do that last time, and I was like, this is broken! And I'm like about to break my computer, and I decided not to do that. So that's good. And then what you do is then you follow the procedures. There's procedures in the study guide that we have here in the class that we're going to be going over. But then you want to make sure you follow your student guide, and it'll tell you exactly what to put in each one. So example, test tube 1 will be 10 milliliters of water. Test tube number 2, you would take your Pepsin. And so you take your Pepsin, we're going to go over here, and we have to then fill up. You have to go back and forth. Come on, Turbo. There you go. And we're going to get a total of 10, because that's what it's asking for. So this is really boring, and you just keep doing this. This is boom, boom. And so we have our 10. All right, good for us. We pick it up, go over here, we put it in the one that is actually named that, which if you want to zoom in and look at it, you can. You just zoom in, and then you move over, and then down. And you can see what it's actually supposed to be named. But you follow your student guide and go all the way across, and you'll be able to actually then see certain things taking place in each one. You give it the time you need. You can finish the rest of this lab actually now on your own, now that you know how to grab everything and put it away and whatnot. And so you just continually basically just follow through with it. We'll stir it right there and put it up and put it back. Oop, used, because if you don't do that, it doesn't work. And there you go. Make sure in between each one you come over, and if you use this, you want to make sure you rinse it out so you're not contaminating each of the, and then put here. And we're good. All right, so people on the internet, just so you know, we're going to go, you can go ahead and now complete this if you would like. I'm going to cut you guys loose so you can go figure that, just so you know, you can't do the essential question unless you do the lab. So either if you have it done already, good for you, you have the answers. If not, go ahead and take that time to finish it up right now. Go in and get it done. And what we're going to do is we're going to actually complete that here in class and knock it out. Good for us. Okay, so I'm going to go back to here. I'm going to go ahead and stop sharing. Boom. There you go. Again, the essential question, how do each of the test tubes react with the egg? Now you know what I mean by the egg and where the test tubes are. How do they react, or why do they react in the way that they do? And to give you some hints and some pointers, is that there's going to be some of the ones you're going to put in that are going to be bases, and some are going to put in their acids, and you're going to see how each of them react with protein, basically. All right, and that is it. I will go ahead and let you go, Jasmine, and you guys can go off and enjoy the rest of your day doing the lab on your own.